Recombinant DNA, Chromatography, and Biotechnology by Laureano Andrade Vicente and the MCAT disciples. Here's the AMC topic list that we're going to be going through. Crossed out a few things that we're not going to go through, and I encircled these. So we're not going to go through distillation or extraction or racemic mixtures. Well, I guess we can go through this in chromatography, but let us get on with it. The scientific method is question, hypothesis, study design, measurements and data collection, data analysis, and conclusion. Question. You need to form a specific question, and you need to form that question after you have done background research on the topic at hand. Once you form a question, you can then formulate a hypothesis. A hypothesis should be testable. You should be able to prove or disprove your hypothesis. So you have to carefully formulate your hypothesis such that that is true. Once you formulate a hypothesis, you have to design a study in order to either prove or disprove your hypothesis. You design the study, you run the study, you take your measurements and your data collection, you analyze the data that you've collected, and then you make a conclusion based on the data that you've analyzed. Very simple. You have to know that. Now what we'll do next is we'll go through a scientific paper written by me, and we will go through it, and you'll notice that it's formulated just like this, because the scientific method is the language that researchers use to describe science. And so it has to be written this way. It doesn't make sense any other way. So we'll go through it. I will read it to you. However, you need to pause this, you need to read this, you need to understand this, because this entire lecture is going to be based on what I did to get from where I started to where I ended. The recommendation that I have for you when you pause the video is that you pause it, you read it, and then you do the passage outlining method that I have showed you in the research-based passage lecture. Okay? Alright, the abstract. The location of a protein-coding messenger RNA, mRNA, in the cytoplasm is actively chosen. However, the study of mRNA trafficking is relatively new owing to the limited technology to allow for the study of this dynamic process at high temporal resolution. Okay, background research. The objective of the study is to demonstrate the ability to override typical mRNA trafficking via the tethering of beta-actin mRNA to the cytoskeleton. Okay, that's what the study is about. To do so, the study utilized a recombinant protein to manipulate mRNA trafficking in immortalized neural cells and subsequent use of fluorescence microscopy to visualize changes in beta-actin mRNA trafficking. Alright, thus methods. Visualization of beta-actin mRNA revealed ectopic tethering of it to actin filaments of the cytoskeleton. This data, hence, mRNA movement can be halted via the expression of constructed proteins as conclusion. That was data analysis and data. And then we had conclusion. This implies that pathways of beta-actin mRNA may be overridden in vivo and could be a target for novel therapeutic approaches to diseases associated with the overproduction of locally translated proteins. That's implications. Okay. Now, we have to go through this as well, because this is essentially what we're going to be going through in this lecture. We're going to be going through what I did as an undergraduate in the lab. And that'll be your segue point into understanding research-based passages. Gene expression is facilitated through the use of a messenger RNA, mRNA intermediate between the genetic storage location, DNA, and the intended gene product, protein. mRNA is not a passive participant in the process of gene expression, and the regulation of this template for protein synthesis provides cells a good opportunity to regulate the production of the gene product. The location of the mRNA within the cytoplasm has the ability to influence the function of the gene product, and therefore the trafficking of mRNA within the cell is one aspect of mRNA regulation at the cell's disposal. The study of regulated mRNA movement, mRNA trafficking, has been limited by a lack of technology that allows for the study of the dynamic processes of mRNA trafficking in real time. However, the advent of coupling of fluorescent markers to proteins that has been adapted for the study of mRNA trafficking. mRNA can move or traffic throughout the cell passively by diffusion or actively through the action of mRNA binding proteins, molecular motors, and cytoskeletal tracts. In order to regulate the movement of mRNA within the cell, RNA binding proteins bind to an mRNA localization element that accounts for mRNA localization and may have both structural and nucleic sequence specificity, forming what then is called a ribonucleoprotein complex, or an RNP complex. This newly formed RNP complex can then interact with subcellular structures directly or act as cargo, binding to the cargo binding domain of molecular motors such as kineses and dyneins, whose motor domains are bound to polarized microtubules. And finally, This motor domain utilizes the conformational chains associated with ATP binding, hydrolysis, and release in such a way as to produce directed movement. That's mRNA trafficking. The directionality of this movement is dependent on the particular molecular motor, kinesins being primarily plus N directed motors, and dyneins minus N directed. The main function of trafficking mRNA is thought to allow for the local translation of proteins. With that in mind, one can imagine how loss of function can negatively impact the cell, as the locally translated proteins play vital roles such as signal reception and organelle biogenesis. The objective of the study is to demonstrate the ability to manipulate mRNA trafficking via tethering of beta-actin mRNA to known components of the cytoskeleton. This was done via the expression of an exogenously introduced plasmids designed to express a RNA binding protein tag fused to a cytoskeletal binding domain together with a fluorescent reporter domain as shown in figure 1. Okay, so what I had to do, I had to create DNA. Okay, I had to make some exogenous DNA, some DNA that is not made normally. I had to make that DNA that codes for a protein that binds to beta-actin mRNA, but will also halt the movement of beta-actin mRNA. And how I chose to do that was taking a cytoskeletal binding protein and coupling it to an mRNA binding protein, which would then tether the mRNA bound to then the cytoskeleton as shown here. So here is the cytoskeletal binding protein, here is the mRNA binding protein. You'll notice that it has a fluorescent marker attached to it, a reporter domain, a reporter gene rather. This protein is a fluorescent protein that will fluoresce upon illumination with ultraviolet light. Okay? And the reason why we need this reporter protein, this fluorescent protein, is we need to know whether or not the mRNA was tethered or not. If we don't have a reporter domain, we can't tell. If we have no way of seeing the mRNA, we can't tell. So we need something to allow us to see it. And the way we allow us to see it is this fluorescent protein. Hence why it's called a reporter domain. It reports on the situation. There's something else that's going on here, which I did not mention in the introduction because I mentioned it further down in the introduction. I just didn't want to make you read too much. There's another protein called MCP-YFP in this cell, being expressed in the cell. Now, this is the construct that was introduced into the cell before I did my experiment. Before I did my experiment, somebody introduced this MCP-YFP, which binds to beta-actin mRNA and will fluoresce. So that tells us whether beta-actin mRNA is present and whether or not it's moving, right? So the idea is that this MCP-YFP will bind to moving beta-actin mRNA and fluoresce and tell us that it's moving. And this construct here, the MCP cytoskeletal binding protein, will bind to beta-actin mRNA on one end, and on the other end bind to the cytoskeleton and stop the movement. So that was what I had to do. Another thing to note, that these MBS stem loops, these are the binding sites for the MCP protein, the coat protein. I haven't defined these things for you yet, but they are defined here. This MCP protein binds to the MBS stem loop. So MCP stands for MS2 coat protein, MBS stands for MCP binding site, or MS2 coat protein binding site. These are not normally found in beta-actin mRNA. This was introduced in a different experiment so that these MCP proteins can bind to the MBS stem loops. What I'm going to go through today is the process to getting from having no DNA at all to constructing DNA to expressing them in the cells and then eventually, live image them and see what's going on within the cell. So let's get into it. Before we can do that, however, we need to go through some units. So 100 nucleotides, NT, 100 NT just means 100 nucleotides. 1,000 base pairs just means 1 kilobase pair. So you may just see KB, you may see KBP, that just means 1,000 base pairs, okay? As you know, the number of nucleotides, the base pairs, is an indication of size, right? The more base pairs you have, the longer you are. So molecular cloning. Essentially what I had to do in my undergraduate laboratory days was molecular cloning. So that's what we are going to go through today. I'm going to go through each little piece that I had to go through in the lab, and I'm going to explain why we did it and how we did it. Molecular cloning is a set of techniques used to insert recombinant DNA from a prokaryotic or eukaryotic source into a replicating vehicle such as plasmids or viral vectors. The vector, the molecule, is introduced into target cells and replicated over and over. Cloning. That's where the name molecular cloning comes from. Okay, so you take a vector, you insert your inserts, which are these little boxes here, and then you express them in the target cell. And that target cell will replicate it over and over and over because it'll undergo binary fission and do its cell division, And with each cell division, you'll have a replicated plasmid. Every time it does DNA replication, you get new plasmid. So that's how that works. That is how you get a large number of your vector. The vector is just a vehicle for your insert. The vector is what will be taken up by your target cell. And in doing so, in taking up that vector, it brings with it the insert, the gene that you want to be expressed in that cell. That is what a vector is. A vector can be a plasmid. It can be single-stranded DNA, it can be double-stranded DNA, double-stranded RNA. It's just the vehicle in which your target, your gene of interest, travels on. It's the passenger in the vehicle. Here are the steps that I had to go through to live image my cells. To live image my cells and to see what the mRNA trafficking pattern was in the cells that I expressed my insert in. The first step is to obtain the target DNA. So we will go through that first. Now, what do I mean by obtain target DNA? Because I told you I had to make the DNA. Well, how I make the DNA is I first start off with the DNA that is normally found within the cell, the neural micelles, and I obtain the gene that I want, namely the cytoskeletal binding protein gene. But as you know, I don't want just the cytoskeletal binding protein gene. I want to attach some things to it, I want to remove some things from it, so I need to obtain my target DNA and then modify it using molecular cloning techniques. So how I'm going to do that is as follows. I'm going to take a pregnant mouse, a pregnant mom. I'm going to do a cesarean section, a C-section on its stomach, and take its prenatal mice, its P0 mice, so they haven't been born yet. And I'm going to remove their brain, and I'm going to lyse their brain cells and extract the mRNA from those brain cells. I don't really want to go into how you lyse a cell. There are a number of ways to lyse a cell. You can lyse a cell via centrifugation, very rough centrifugation. You can lyse a cell via detergents, very rough, harsh conditions. All right, let's just say that I lyse the cell. The next step is to extract the mRNA from those cells. So let's do that. Let's show you how we do that. Before I can show you how to do that, I'm going to have to go through chromatography. So I'm going to give you a primer on chromatography, and then eventually I'll show you how I extracted the mRNA, the specific chromatography experiment that I had to do to get the mRNA from the cells, so in the cell lysate, so the lyse cells, what was in the cells. In any chromatography experiment, there will be a stationary phase and a mobile phase. Different components of the mobile phase will move at different rates according to their affinity to the stationary phase and solvent, allowing for their separation. Pretty simple. I'm sure you're familiar with chromatography. Here's paper chromatography. I'm sure you've done this experiment. Paper chromatography can be used to separate and identify compounds based on their RF values. So here is the setup for a paper chromatography experiment. You have an enclosed apparatus, and within the enclosed apparatus you have a membrane, the stationary phase, and a mobile phase. The stationary phase typically is cellulose membrane, and the mobile phase is just some solvent. Can be polar, can be nonpolar. Typically it's going to be polar. What's going to happen then is you're going to draw a little line with a pencil and dot whatever compound that you have that you want to run on your paper chromatogram. So the chromatogram is this, the paper, the stationary phase. What's going to happen is the solvent is going to move up towards the top of the paper as you keep the apparatus enclosed over time. It'll travel up because it's attracted to the paper. It's attracted to the cellulose membrane. It's a polar membrane. This solvent has to be polar in order for it to do that. It has to be attracted to the cellulose. So as it's attracted to it, it'll move up. At some point, you'll open up the apparatus and the solvent will stop moving. So the solvent front will be marked here. You'll mark it there. And then what was in these little pigments here that you marked onto your chromatogram will move with the solvent front, but they'll move at different rates. And they'll move at different rates because they're attracted to the stationary phase at different proportions. So the more polar a compound is, the more attracted to the stationary phase it will be, assuming that the stationary phase is polar itself. If it was nonpolar, it'd be the opposite, right? The nonpolar would be attracted to it more. That means that the more polar you are, the slower you will move through this chromatogram, this stationary phase. All right? The more attracted you are to the stationary phase, the slower you will move through the stationary phase. Then you can calculate an RF value. So the RF value is based on the properties of the compound itself. It's based on the affinity of that compound to the stationary phase, so it's going to be specific to that compound. and that'll allow you to identify different compounds in a solution where you don't know what compound are in the solution. That's often done at least in intro bio classes with different pigments. The RF value, the distance traveled by the compound over the distance traveled by the solvent. I think that makes intuitive sense why that would be specific to each compound. There's thin layer chromatography. The only thing different between thin layer chromatography and paper chromatography for our purposes is that the stationary phase is the silica or aluminum hydroxide stationary phase as opposed to cellulose. But they both have OHs coming off of them, and they are all attracted to the OH. So the same properties that you know about paper chromatography are also true for thin layer chromatography. So the more polar compounds will be attracted to the silica, and the less polar compounds will be less attracted to the silica and move more quickly. So this would be, let's say, a less polar compound. This would be a more polar compound. Then it has column chromatography. This is much more useful and interesting to us as biochemists. All right, there's a bunch of different ones that we can talk about. There's gel filtration chromatography, there's affinity chromatography, there's ion exchange chromatography, and there's hydrophobic affinity chromatography. Let's go through each one. So gel filtration chromatography. I've gone through this before, so I'm going to do this rather quickly. In gel filtration chromatography, you put these little gels in these gel beads in the column. Within each bead, if you zoom in onto the bead, you'll find that there are little passageways for molecules to pass through. However, they have to be of a certain size to fit through the passages. Now, it turns out in gel filtration chromatography, the smaller molecules take longer to elute from the column, and the larger molecules take less time to elute from the column because they cannot fit in these little passageways. You can imagine that each one of these beads have these passageways. So the big molecules can pass right through these beads and go right through the side of it and empty out into then your container, where the smaller ones have to travel through all these little passageways, and as soon as they get out, they get into another bead. As soon as they get out, they get into another bead. As soon as they get out, they get into another bead. So they can spend a lot of time going left and right instead of up and down. So they take longer to get through the column. That is what gel filtration chromatography does. It allows you to separate on the basis of size. You have affinity chromatography. So affinity chromatography just takes advantage of the fact that protein interactions are specific. In order to extract your protein from a mixture of a bunch of different other proteins, you can attach a binding protein that will bind to your protein of interest to then the column, to the resin, the solid resin. So a resin, if I haven't told you before, is just a solid matrix, just a very dense solid. So what you do is you attach a binding protein that will bind to your protein of interest to the solid resin, and then it'll separate. And when you run the flow through, when you run your solution through the column, it'll bind to your protein of interest and separate it from anything else that's in solution. A number of ways you can do that. You can use antibody that'll bind specifically to its antigen, or you can use, for example, a nickel NTA resin that'll bind to polyhis tags. How do you get a polyhis tag onto your protein of interest? Well, you use molecular cloning and insert codon that'll code for histidine residues in your protein of interest, such that at the end of your protein of interest, at the tail end of your protein of interest, there'll be polyhistidine residues there that can bind to then the column. That's a great way to essentially non-specifically bind your protein of interest. So every time you make a protein, you just add a polyhis tag into your vector DNA such that it'll express your protein with a polyhis tag at the end, so you can separate it out from any other protein in solution. You may not appreciate what that means now, but hopefully by the end of this lecture, you will know what it means to add a polyhis gene, a bunch of histidine codons, to the end of your gene, of your DNA of interest, such that it'll express it with polyhis tag protein that can be bound by a nickel NTA resin and separated from the rest of solution. Now, it just so happened that polyhistidine residues bind with very tight affinity to nickel ions. You wouldn't be able to know that if I didn't tell you. You can have ion exchange chromatography. So let's get this straight right here. In anion exchange chromatography, the resin binds anions. That means it's positively charged. In cation exchange chromatography, the resin binds cations. That means that the resin is negatively charged. Okay? Obviously, they'll separate from the rest of solution. And then you have hydrophobic interaction chromatography, where the column is hydrophobic And so hydrophobic molecules via the hydrophobic effect will move more slowly through this column than hydrophilic compounds. And it'll separate then on that basis, on the basis of hydrophobicity. Hope that all makes sense. I've gone through it before, and this is just a review. So what I had to do to get my mRNA from the mouse cells was as follows. I had to take advantage of the fact that mRNA has poly-A tails. So I took advantage of the fact that I can use a filter that had an oligo-DT probe that can specifically bind to the poly-A tails of mRNA. Now recall, the only nucleic acid in the cell that has a poly-A tail is the mRNA, because we do not get the poly-A tail from the template sequence of DNA that we transcribed to get mRNA from. That doesn't have a poly-T to get a poly-A tail. No. What happens is, during transcription, there'll be a cleavage event after passing an A2UA3 sequence, and there'll be a poly-A polymerase to go and polyadenylate the 3' end of the nascent mRNA. And that's how we get the poly-A tail. And so that's why the mRNA is the only nucleic acid that has poly-A tail, and we can utilize that to then selectively remove mRNA from the mouse extract and not any other nucleic acid. So what I did is I nestled the filter tube in a centrifuge tube, and I centrifuged it and ran this mouse extract through the filter tube. And when it ran through the filter tube, the oligo-DT probe, the short deoxythymidine probes, bound to our mRNA. All the mRNA that was found within the mouse extract. And there were still a few impurities in there, so what I did next was I washed the tube a little bit, and I added the elution buffer after that, and I switched the centrifuge tube to a clean centrifuge tube so that when I elute or remove the mRNA from the filter and enter it into then the centrifuge tube, it would be clean, and it would only contain mRNA. So I elute it, and hell, look at that. What I have now in my tube is only the mRNA from the sample cells, and it has every single mRNA because every single mRNA has a poly-A tail associated with it. Again, the reason why I'm doing this and the reason why I'm obtaining mRNA is because I have to obtain the sequence of the gene that I want to eventually splice with some other genes to make my construct. And I don't want any introns within that gene. I do not want any introns because I'm going to express it eventually in neural mice cells, and one of the last things that you want is a different intron splicing pattern that would occur in the mouse cell such that your protein that gets expressed isn't the right protein. So you want to remove any introns. Make sure you only have exons. Okay, so I extracted the mRNA. The next step is to convert that mRNA into DNA, cDNA really it's called, but it's just normal DNA, that I can then splice up to get and make my construct. So let's do that. We have to do reverse transcription to get from the mRNA to cDNA. Okay, so let's do it. As you've probably seen this slide before, the process of doing reverse transcription in vitro, that is in the laboratory, is called RT-PCR, or reverse transcriptase PCR. What you do is you take an oligo-DT primer. Again, you're taking advantage of the fact that mRNA only has a poly-A tail, or all mRNA has poly-A tails, and you're amplifying that via RT-PCR. It's just PCR, but it's using reverse transcriptase enzyme. You use the NTPs, you add magnesium chloride, all this jazz, a buffer solution, and you make cDNA. Again, as you know, cDNA has the following properties. It is complementary to the mRNA it was copied from, and it contains only exonic sequences. Again, I only want exonic sequences. I only want sequences that are going to code for protein, because when I introduce this DNA into another species that may not have the same splicing machineries that our eukaryotic normal human DNA has, so we want to make sure that our protein that gets expressed is going to get expressed. It's not going to mess up because of some intron. cDNA can be incorporated into a vector plasmid and transformed into bacteria to ultimately be used to form a DNA library. A DNA library is the set of bacteria who replicate vector plasmids with the cDNA of interest, allowing for long-term storage and accessibility when needed. However, we're not done yet. So that was RT-PCR. All right, and I haven't talked about PCR yet. I'm going to talk about PCR next slide. What we need to do now is take that cDNA and amplify it and get a complementary strand to it. Because right now, we only have one strand of cDNA, but we don't have a complementary strand to cDNA because we can't make a complementary strand because reverse transcriptase only works from mRNA to DNA, not the other way around. So we need to do PCR to do that. We need to introduce some new polymerases to run that. So then the next step is to PCR the target sequence of cDNA. Again, we need to make the complementary strand. So here we are at PCR, polymerase chain reaction. So PCR is the technique used to amplify DNA into millions of copies of itself. It's a laboratory DNA replication method. As you know, DNA replication occurs within the nucleus. However, if we want to do it in the laboratory, we need another way of doing it. That other way of doing it is polymerase chain reaction. So it uses the same and follows the same properties of that of in vivo DNA replication. You need a polymerase and you need primers. You need a template DNA strand and you need the NTPs to be incorporated into then the growing polymer. So that's what all these typical components are about. You'll notice you don't see magnesium here, and that's because it's incorporated into the Taq reaction buffer. Alright. There are 3 steps to PCR, truly. 3 real steps in the cycle of PCR. It's denaturation, primer, annealing, and extension. The denaturation is handled by increasing the temperature of the reaction mixture in the thermocycler. Okay? That is in order to separate the strand. In vivo, what separates the strand is MCAT27. It's the DNA helicase. However, we don't have that in vitro in the lab, so we need to do that by increasing the temperature. The temperature increase denatures the DNA by increasing the kinetic energy of the molecules. By increasing the kinetic energy of the molecules, it provides enough energy to break up the hydrogen bond that keeps the DNA annealed to one another. So that denatures it. Alright? Now our primers can now bind to the denatured strands. Alright? That we decrease the temperature down to a temperature where they can bind. There's some nomenclature associated with this temperature. The TM, or the melting temperature, is the temperature at which 50% of the primer is annealed to the template strand. The primer will only anneal to the temperature strand around that melting temperature. And no, it's not actually melting, that's just the terminology. And annealing just means to hydrogen bond and pair up with the template strand. That's what annealing means. Another way to say that is hybridize. You may also hear the term a primer tail. So a primer tail, the tail of nucleotides that do not bind to the template sequence and are used to add on DNA to a sequence. The reason why we would ever have to use a primer tail is that typically when we want to amplify a gene to eventually introduce into another cell, we need a way of cutting that gene in specific places such that we can incorporate it into the vector without damaging the gene. The way we do that is we add a sequence of nucleotides, a restriction site, to the ends of our gene. And we do that via primer tails. You may not understand what that means yet, but we'll get there. So a primer tail would look just like this. It would be like this, and it won't bind to the template DNA. However, what will happen is, and I'll show you this later, a polymerase will come and fill this in, and it'll fill it in this way. So at the end of the replication thing, you will see that you'll end up having then this thing here, and then DNA polymerase will also replicate this way and replicate the template strand. And what you'll end up doing then is adding on a sequence of DNA. So we'll show you that next. So the next step is extension. Polymerase will go and extend. And a polymerase will do this at a certain temperature, around 68 degrees Celsius. I mean, there's nothing special about that. You know that, different temperatures will cause for different levels of denaturation of protein, and turns out Taq polymerase works at this temperature. Taq polymerase is a special kind of polymerase. And it's a special kind of polymerase because it came from an organism that lived in very hot environments, and so it does not denature completely under conditions of high temperature. A lot of proteins, when they get denatured at very high temperature, they are irreversibly denatured. Okay? There are some proteins that aren't irreversibly denatured, but some are. And it turns out most polymerases, like our normal DNA polymerase, will become completely denatured under high temperature and won't be able to revert backwards. The Gibbs free energy of its denaturation is negative, and the Gibbs free energy of its renaturation is positive. It's not spontaneous. But it turns out Taq polymerase can withstand high levels of temperature without being completely irreversibly denatured. So that's why we use Taq. And that is PCR. You'll do that for many different cycles, and you keep replicating the DNA. If after cycle 1, that's what it looks like. We double the DNA. Cycle 2, double the DNA. Cycle 3, double the DNA. Cycle 30, you get 2 to the 30 DNA molecules. That's a lot of DNA. The reason why we do this is, again, to obtain our target DNA and to get it in enough concentration that we can extract it from the rest of the stuff in solution. And the other part of it, the reason why we have to use the primer tail, so I had to use the primer tail. And I had to use the primer tail to add restriction sites so I can cut my gene of interest and insert it into the vector, which we'll get to soon. So there are some rules about designing primers that I should go through here. The first rule, that primers must be written in a 5' to 3' direction. That's going to be our most important thing out of this slide. The two primers must be pointing towards one another, flanking the region you want to amplify. And the primer should be 20 to 30 nucleotides long, giving a TM, a melting temperature about 60 degrees Celsius. Again, the TM is just the temperature at which 50% of the primer is bound to the DNA. The annealing temperature should be at least 5 degrees Celsius lower than the TM of the primers. So all that does is make sure that the primers bind to the sequence. Of course, you want the primers to have a very close to the same TM so that they would both bind around the same temperature. Otherwise, you can't really run the PCR experiment, because these rules here, these two rules, just result from the fact that you need to amplify a sequence of DNA. In order to do that, you need primers. And that DNA, they must be going in the same towards each other. Otherwise, you'll end up replicating things that won't base pair with one another. So you'll get a taste of that in the next exercise here. The exercise is for you to design a pair of 8 nucleotide primers that will only amplify the blue region of this DNA map of insulin cDNA. Go for it. Okay, so here are the answers. The forward primer would be 5' ATG GCCCT 3', and the reverse primer would be 5' CTA GTT GC. Now, if you didn't get that right, let's explain this to you. This DNA map only showed one sequence. It only showed one strand. It doesn't show both strands. And that is typically what you will find when you look up DNA maps of genes. It just makes it simpler for us as biochemists to just look up one strand, because we know the complementary strand looks complementary to this strand. So that's what you'll find if you look it up on any of those websites, those databases like NCBI or something. I told you that the primers have to point to one another. Okay, and you write them out 5' to 3', and they bind to the sequence and they point towards one another, and polymerase extends from 5' to 3'. So the forward primer does not bind to this sequence here. You'll notice it's the same sequence as what's written here, but it does not bind to the sequence. It actually binds to the complementary sequence. It just looks like this because it binds to the complementary sequence. Now the reverse primer has to bind to this sequence. Okay, it has to bind here. And so it has to be the reverse complement of this, because this is running from 3' to 5', GATCACG. So it has to be the reverse complement. So you have to write from 5' to 3', it has to be C. Okay? It has to be T. It has to be A, because it's complementary. And it has to be running in the right direction. Okay, I think you can see the pattern here. And you'll see, if you write this out yourself, you'll notice that then, okay, yes, this is complementary to this sequence here. And then this primer is complementary to the sequence that is not written down on this page. Alright? You have to just deal with that and figure that out, because I'm telling you, but you probably don't understand because you haven't written it out and kind of just dealt with the issue at hand. Alright, so we did the PCR of our target cDNA, and we did add on PCR, so we actually added on sequences to our gene of interest in order to then later be able to cut it and add it to our vector. So now our next step is to extract and purify our cDNA with a non-specific column chromatography kit. So in order to do that, we need to do a gel electrophoresis experiment, actually an agarose gel. And the reason why we have to do an agarose gel is because we need to separate our cDNA from any of the other cDNA that's in the solution. Because when we did RT-PCR and then regular PCR to amplify the mRNA that we had, we amplified all the mRNA into cDNA. We want to separate our cDNA from any other cDNA that's in solution. The way we do that is with gel electrophoresis. It's a separation technique, which I've already gone through how gel electrophoresis works, so I'll go through this rather quickly. It's a separation technique. Alright, so we separate. We turn the battery on. Remember that the cDNA and RNA and anything else, the nucleic acids are negatively charged, so they are repulsed by the negative electrode and attracted to the positive cathode, so they move according to size or according to length and move down that way. So a larger thing, they're up here, smaller ones up here. If you take a cross-section, this is what it looks like again. Alright, it's cross-linked cellulose polymer and form the molecular sieve and separates on the basis of size. Larger sizes move slower, smaller sizes move faster. I think it's pretty obvious. Larger you are, the more negative charge you have, but the harder it is for you to move through this cross-section of this cross-linked polymer. So clearly it'll take you longer. Now, let's say our cDNA was this one right here. What we do is we cut out that band, our amplicon, okay, and put it into the centrifuge tube for purification. With a gel extraction, you use the filter and a series of centrifugations to separate DNA from the agarose gel. At the end of the gel extraction, you have purified DNA. So let me show you that. What you do is you cut out the little band in the gel. This gelatinous, it literally feels like jello. What you do is you take a razor blade and you cut it out, a little piece of the jello out that contains your DNA, your cDNA, and you solubilize that jello. Okay? Then you add it to the centrifuge tube. You add it to a filter tube that is nestled into a centrifuge tube. You centrifuge it, you filter it, and then the nitrocellulose matrix will bind to then your cDNA. You wash it a little bit, and you add the elution buffer, and you elute it out into a new centrifuge tube, and then you have your purified DNA. The nitrocellulose structure is shown here. Now, it's not clear why nitrocellulose binds nucleic acids. And it's not that I just don't know how it works, it's just nobody really knows how the binding mechanism works. But what is clear that nitrocellulose will bind non-specifically to nucleic acid. And so that allows you to separate nucleic acids from anything else that might be in solution. And some of you might be wondering, if you're paying attention, how do I know that it is my DNA of interest, that was bound to the nitrocellulose matrix and not just any old DNA that's in solution? Well, because the reason why I did the gel electrophoresis was to make sure I only have my DNA in solution. That's why we did the gel electrophoresis before we did this experiment. Because if we did this column chromatography experiment before we did the gel electrophoresis experiment, we would just bind all the DNA that's in solution, not just our target DNA. And that's why we had to do the gel electrophoresis experiment first. So what we have in this solution is just my purified DNA and just the gene that I want to link up with other genes, plus the add-on PCR sequence that I had done with the add-on PCR. But that's my gene, I just modified it a little bit. Alright, so we've done a lot of the battle. We obtained our target DNA, and we did that by extracting neuromicelles, this mRNA from neuromicelles, using column chromatography, and then we amplified it. We did RT-PCR, reverse transcriptase PCR, to get cDNA, and then we amplified the cDNA via regular old add-on PCR. And we call it add-on PCR because we added on a little bit of sequence of the target, a little bit of sequence DNA onto the end of the target DNA using a primer tail. We extracted that cDNA. Now we have purified cDNA. Now what we want to do is we want to construct our experimental construct. So we want to cut it up and make sure everything's bound up to one another. And then we want to do the same thing with the vector DNA and insert into the vector DNA. That's what we're going with next.